It's an honor for me to be here and present my PhD work that I did in Tobias Bonhoeffer's lab in Munich. And I'm going to talk about neural representations of learned rules for categorization in mouse prefrontal cortex. What do I mean with learned rules for categorization? Categorizing things refers to sorting them into groups, and we often learn or form rules to do that type of categorization. Let me give you an example. Take music. We can sort a song into, for example, a playlist based on the genre that it belongs to. So we can have classical songs, we can have jazz songs, hip-hop, and so on and so forth. But we can also sort a song based on the mood we're in when we like to listen to it. And so very flexibly, we can sort the same thing into two completely different categories dependent on what rule we apply to it. And this is what I mean when I say rule-based categorization. In a research setting, this has been explored in humans, for example, by asking subjects to categorize letter stimuli based on their font, or their color, or their size, and we can very quickly iterate through different rules. And parallel fMRI recordings have shown an increased activation of several prefrontal cortex regions while human subjects were performing this type of categorization. So that indicated an involvement of that brain area. Studies in non-human primates could then add a lot of detail to what might prefrontal cortex be contributing to that type of behavior. For example, a study by Earl Miller's lab has trained monkeys to categorize images of cats and dogs based on two different schemes or rules, one discriminating between cats and dogs, and one discriminating between two prototypical instances of dogs or cats. And then electrophysiological recordings in prefrontal cortex, here dorsolateral PFC, has described category selectivity. That is a response property of individual neurons that fire only in response to stimuli of one of the two categories, like this example neuron here. And for both of the schemes, a fraction of neurons, between 10 and 20%, have been described to show this type of selectivity. However, when the authors compared category selective neurons from one scheme to the other scheme, so plotting the response property of this example neuron when the monkey is performing according to the other scheme or rule, they found that that neuron was not selective for the other categories. And in total, when quantifying the overlap between those two populations, they found a chance level of overlap if you independently sample twice from the same distribution. So they could conclude that prefrontal cortex neurons represent categories in a rule-dependent way. This was the stage when I started my PhD and we decided we want to add the mouse model to this type of research. One, because it's interesting for comparative reasons, and two, because of the circuit interrogation and investigation tools that the mouse offers. But before we could get there, we had to answer two questions, and those I will focus on today in the talk. And the first question is, can mice learn these types of visual categories? And the second question, and this is what I'll focus on since we're in a behavior session, but then I'll also briefly go into, does mouse PFC represent these learned categories? And does it do that in a maybe similar way to what we know from the primates or in a different way? And then I want to spend 1 minute in the outlook on two projects or two questions that we can now answer, where we can start to use the mouse model. What can we learn beyond what we know from primates? All right, let's start with the behavior. First, we had to come up with a visual category learning paradigm in mice. And the way we went about this is by training mice in a setup that looked like this, where the mice were head fixed and on a treadmill and were water restricted to stay engaged in a go-no-go task. That meant that they had one lick spout in front of them and could decide to lick or to withhold from licking in response to seeing a visual stimulus on the monitor in front of it. The visual stimuli were drifting gratings, so simpler than the cats and dogs used in the primate studies. But similarly, we spanned a stimulus space by varying the orientation and the spatial frequency of the stimuli. And then we divided this space up into two categories, either following the orientation or the spatial frequency, and those are our two rules. So following, for example, the spatial frequency rule, we'd end up with one rewarded and one non-rewarded category. The way we evaluated the behavior of the mice was by looking at their choices per stimulus. So the way I'll plot the behavior of mice is by plotting every stimulus as a pixel and its color indicating the fraction of go choices that a mouse made in response to seeing that stimulus. So let's look at one example session of one example mouse before it learned anything. This is a time point that will be much more crucial when we come to the neural data, but here you can familiarize yourself with the plots. Each pixel here is one stimulus, and the mouse doesn't discriminate between the stimuli. Then we started training the mice, but we didn't throw all the stimuli at the mouse at the same time because we were afraid that it's too complicated, so we started training the mice with only two stimuli, the easy ones far away from the category boundary, until the mice were familiar with the task scheme of go, no go. And only then we gradually introduced more stimuli to each category, first to 3 per category, and then to 9 per category. And now you can see for this example mouse that it nicely distinguishes between the rewarded category and the non-rewarded category. But one of the hallmarks of rule-based category learning is the ability to take what you've learned, the rule, and to apply it to novel situations, novel stimuli, just like what Andreas just talked about. It's generalization. So we wanted to test our mice for this, and we did that by, after each mouse had learned this stage, presenting in one session the second half of the stimulus space, and comparing the performance on novel stimuli to the performance on experienced stimuli. And what we found was that the mice could do that just fine. Now, this is an example mouse and the average across a whole session. So you might say, well, haven't the mice just learned to integrate stimuli very quickly upon receiving feedback? In order to address that, we can look at the very first presentation of every of those novel stimuli and see, does the mouse do it correctly before receiving feedback? And this is the data for this example mouse, but also here as D prime for all mice, we didn't find a difference between novel and experienced stimulus performance. And that led us to conclude that mice really can generalize learned rules. Another hallmark of rule-based categorization is the rule switching. We've already talked about it before. And our stimulus space allowed for that too. So after a mouse had learned the first rule, we switched the rule that applied to the stimulus space, and we required the mouse to recategorize the stimuli based on a visual feature that they had to ignore before. Also here, we didn't present all the stimuli right at the same time. We gradually built up the category from 2 stimuli up to 9 stimuli again. And what we found was that they could learn the second rules. That allowed us to test for generalization again. And now there was an interesting opportunity here, because those novel stimuli weren't really novel. They had experienced them with a different rule before, so they had learned a different type of behavior. So we could compare, do the mice prefer to generalize a rule, or do they prefer to rely on prior knowledge? And when we compared the performance of experienced and novel stimuli, we found, again, no difference, indicating that the mice prefer to generalize that rule rather than to rely on the prior knowledge. And with that, we concluded that mice can switch rules for categorization. And we were happy with the characterization of this category learning behavior, so we moved on to characterizing the neurons in prefrontal cortex. And the method of choice for us was 2-photon calcium imaging. And here we focused on prefrontal cortex, and specifically the areas anterior cingulate and prelimbic cortex. And since both lie, or the majority of that piece of cortex lies within the hemispheres of the mouse brain, we had to come up with a trick to make it accessible to photon imaging. So we implanted microprisms in between the hemispheres to image around the corner. And together with transducing neurons specifically in layer 2-3 with 2 fluorescent proteins, one a functional protein, a calcium indicator GCAMP, and one a structural activity independent protein, we were able to track the very same neurons through our behavioral paradigm that was like 2 to 3 months. And that allowed us to quantify stimulus or task-related activity in individual neurons of naive, learning, and trained mice, and even follow them through the rule switch. The first thing that we looked for in our data was category selectivity, because we had a strong expectation from the primate results that prefrontal cortex might represent these categories. And in our data, category selectivity would mean that a neuron responds to either the top or the bottom half of the stimulus space. And so when we looked for stimulus-evoked activity in individual neurons, we found exactly that. Here again, each pixel corresponds to stimulus-evoked activity of one stimulus, and this neuron responds to the top half of stimuli, whereas I can show you another neuron that responds to the bottom half of those stimuli. Now, of course, those are example neurons. We wanted a way to quantify this type of response behavior across our population, so we calculated a category tuning index that essentially compares response similarities of individual neurons to stimuli across the category distinction and within the same category. And a neuron that showed larger differences in its response across categories would get a positive CTI value. And now, this value we took per neuron, we thresholded it, and we called every neuron above a CTI of 0.1 category selective, and then we started investigating our populations. And the first thing that we observed was when we looked before learning, there was hardly any category selective neuron in prefrontal cortex. However, after learning, as soon as the mouse was categorizing according to a rule, we found about 8 to 10% of all neurons showed this type of response behavior. And this was nicely in line with what we expected from the primate. But we could also add to that this is a learned feature of prefrontal cortex. And now, of course, we could make true on our promise of two-photon imaging. We could follow this population through the learning. And for that, I will split populations of category selective neurons that we identified that way into neurons that responded preferentially to the go category selective neurons, and no-go category selective neurons. And from now on, I will show you averages of this group of neurons to all of those stimuli. And the first thing that we observed was that those groups of neurons really nicely selectively distinguish between the two categories after learning. Now, looking before learning, in line with what I just told you before, there was hardly any stimulus response and no selectivity. And now comes an interesting fact, because when we followed those neurons across the rule switch, we saw that on average, the go category selective neurons changed what they responded to, changed the stimuli that they responded to in order to represent the new go category. And in line with this, when we selected category selective neurons for the second rule and looked at how did they respond before the rule switch, we found some prior selectivity here. And this was in contrast to the no-go category selective group, because they rather lost their selectivity when those categories became irrelevant. And a separate group of neurons built up selectivity. And this indicates a different level of overlap between those two groups, which we can also, of course, directly quantify. Here in the hollow bars, you can see the overlap between go category selective neurons of rule 1 and rule 2, and here for the no-go neurons, and we can compare that to the expected level of overlap given independent sampling. This is the gray box here. And the one thing that we found was that there was significantly higher overlap for the go populations of neurons, but for the no-go populations, this was within the range of independent sampling. And now we do have a strong theory why there's an increased overlap here, and that has to do with other task-relevant representations in prefrontal cortex, so motor planning and reward, which we addressed in an experiment that I don't talk about today, but I'm happy to talk about it in the discussion. But the no-go population and its independent sampling across two rules was very much in line with what we knew from primates. And this leads me to my conclusions of today, where I hope I could convince you that mice can learn visual categories, they generalize and switch rules, and neurons in mouse prefrontal cortex represent these learned categories, potentially in a similar way to the primates. And now, what can we do and what can we learn beyond? There's two questions that we are curious to follow up on. One is the question, now how does PFC build these task representations? And since we now know that there is a representation of learned categories in a trained animal, but it's not there in a naive animal, we have the chance to, with high temporal detail, try and follow these representations while they are emergent and look closely at what changes in firing patterns can we observe during this time period. And the other question is much more behavioral related, and that is we can perturb the system. We can try and ask the question, what specific part of this behavior is PFC dependent, if any? So is it category memory? Is it category learning? Is it generalization specific? And there we can use the convenience of optogenetic perturbation in the mouse to try and answer these questions. And with this, I'd like to thank mainly my advisors, first and foremost Peter, the postdoc that supervised me and did this project with me, Tobias and Mark, who also supported this project in the lab, and our other collaborators, and now you for your attention.